like fits, yeah. I mean, it fits a 32 ounce, but I like my 24 ounce. So I always have it. Yeah, so I always have it when I go somewhere. In my snacks. I'm obsessed. I bought like 3 different colors. Yeah. And then the belt bag, the everywhere belt bag, they're always out.
Oh, that's what I have.
I know, but they're always out. Like I was stuck. Dang, I want a different color.
If you that you show my business, I don't really like some other classes. You already need some.
Okay, so welcome to class, everybody. Does anyone need a chapter 9 packet?
Also, he is recording. Okay. Airdrop me after class. Thank you.
All right, so the plan for today is we are going to keep talking about chapter 9. Towards the end of class, I'm just going to, if you are completely happy with exam number 2 and don't have any questions, you're out of here. And we'll spend the last little bit going over the exam if you're not, okay? Instead of doing it at the beginning. So on Thursday, we'll take quiz number 9. We'll do something afterward. I don't know where we'll get, we'll see. Wherever we stop on Thursday is the last of the material is fair game on next week's exam. So next Thursday, we're going to have exam number 3. Okay. Same exact format. We'll talk more about what's on the exam, what's not on Thursday. And I'll get the learning objectives posted for it. Okay. So any questions on what we're doing this week?
So,
week 11.
Are you going to have 8 and 9 or just 9 on this quiz coming up for?
So it'll be 8 and 9. 7 we've already had. That's exam number 2, right?
Yeah.
Yeah. So chapter 8 and chapter 9.
8, because you just had 9 in. That's only what I have.
This is what we're covering this week. The test is next week.
No, for the text though.
For the test is going to be chapter. Oh, everything we talked about last week. Remember that was.
Okay.
Yeah, I don't remember exactly where we started and stopped, but look in your notes, whatever we covered last week. Okay. So any questions on this week or next week? All right. So in that case, we're going to keep talking about translation. So we're going to start this off by watching the video that we already did, and then we're going to draw some more pictures.
Take back our streets. Treat criminals like criminals. Fully enforce the law and make sure our police have the resources they need.
Who's looking forward to no more political commercials on YouTube?
Prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a 3rd site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the 2nd amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the 2nd transfer RNA is released from the E site. the ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay.
Proposition 132 ends majority rule in Arizona.
Okay, so what we're gonna do is talk a little bit more about what that video is talking about and kind of fill in some blanks. So remember this from last time, here's a bunch of them on here, but they're all the same thing. These are codons. These are codons in the messenger RNA, and this is telling us which amino acid that the codons code for. So some of the codons, there's 4 different codons for the same amino acid. An example would be valine. Any 4 of these codons are going to code for valine. Some of them don't have 4, right? So glutamine here only has 2 codons, right? So these are all the possible codons you could ever have. There are no more than this. If codons are 3 nucleotides long, which they are, and they're made of one of 4 things, i.e. A's, T's, G's, and U's, or A, U's, G's, and C's, then there are no more combinations. So every single one of them is accounted for right here, okay? So if we know what the codons are in a messenger RNA, we know what order to link the amino acids together in. But it's the ribosome's job to do that, right? So a ribosome has to be able to look at the sequence of a codon and know which amino acid goes next. So there has to be a connection between codon sequence and amino acids. And what is that connection are these molecules here. So these are tRNAs, or transfer RNA. So transfer RNA is the link between codon sequence and amino acid sequence. Each tRNA has this general shape. It's nothing but a sequence of RNA, right? So during transcription, we make messenger RNA, RNA polymerase does. RNA polymerase also makes transfer RNA. You see how I drew this kind of this funky shape? This is just one linear RNA molecule that just happens to fold up in a secondary structure that looks a lot like this. So this is the 5' end of our transfer RNA, and this is the 3' end of it. It's just a single strand of RNA, just like messenger RNA, only its job isn't to be a code that the ribosome can decode for a translation. It's structural, okay? So transfer RNAs are nothing but a long linear molecule of RNA that folds up into a structure that looks like that. But there's 2 very important things that a transfer RNA does, right? The first thing that a transfer RNA does is it always carries an amino acid. So we're going to say this transfer RNA holds the amino acid methionine, right? So we know that our start codon is AUG. So there's our first letter, there's our second letter, third letter. AUG codes for methionine, right? So we need a transfer RNA that carries a methionine and can also recognize a start codon, which is AUG. So here is our start codon, right? So if we look at this, let's look at the start codon completely separately for a second. This is our start codon. It's an AUG. So if we were going to have another piece of RNA, a different RNA molecule bind to this AUG, would this be 5' or 3'? It'd be 3, right? DNA and RNA are always anti-parallel. So if this RNA nucleotide was an A, what would this be?
A U.
Be a U.
An A and a C.
So if I had a sequence of 3 nucleotides that went 3' UAC, that would bind perfectly with this AUG. And that's exactly what happens. So this transfer RNA that's carrying the methionine has an anticodon sequence that goes CAU. You see how this is the 5' end of our transfer RNA? I have it labeled. So we follow that along. This is the 5' end of that anticodon. This is its 3' end. So this transfer RNA, which carries methionine, can travel around here. Is it going to bind to this codon? No. Is it going to bind to this codon? Is it going to bind to this one? Absolutely. Right? So here's the thing. Transfer RNAs are the bridge between codons and amino acids because they have what's called an anticodon. Anticodons bind to codons. Now here's the thing. This UAC only binds to this AUG, like we talked about here. So every single transfer RNA period in a cell that has the anticodon 5' CAU always has a methionine on it. It doesn't have a valine, it doesn't have a glutamine, doesn't have a cysteine, doesn't have any other amino acid. So if you're a transfer RNA and if you have this anticodon, you must carry this amino acid. If it was, for example, carrying a valine, which is not correct, then that transfer RNA would bind right here, and then AUG would code for a valine. But we know it doesn't, right? So this is super important. The link between codon sequence and amino acid sequence are these transfer RNAs, okay? So let's see if we can predict a few things. This is going to be a terrible drawing because I'm not an artist. I'm trying to draw the ribosome. So this is the small ribosomal subunit. This is the large ribosomal subunit. That movie talked about small ribosomal subunit, large ribosomal subunit. Use your imagination, right? So the ribosome had 3 different areas in it. What was the one the far left called, remember? E. It's the E site. E stands for exit, right? This one was the P site, right? This stands for peptide, because this is where peptide bonds are being formed. This is the A site. This is the acceptor site. It's called the acceptor site because this is where the next transfer RNA is coming into. So if we have a transfer RNA sitting right here, and it's bound to the codon ACA, what's this anticodon sequence? UGU. UGU. Is this the 5' end or 3' end?
That's the 3'.
5', anti-parallel, right? So if that transfer RNA has a UGU 3' to 5', it's going to base pair with the messenger RNA, right? Because this is going 5' to 3', this is going, the anticodon is going 5' to 3' the other way. The base pairing rules are being followed here. What amino acid must this transfer RNA be carrying? What?
Amino acid?
ACA, right? What amino acid does that codon code for?
Threonine.
Threonine, right? Yeah, sorry, these are like tongue twisters. So we know from the chart that if we have ACA, that codes for a threonine. So this amino acid has to be bound to a threonine. So if I give you a sequence of RNA and draw a transfer RNA right here, and that's it, you'll have the chart, of course. You can tell me what the anticodon sequence has to be. And you can tell me what the amino acid has to be, okay? Now, this is the 3rd codon in this messenger RNA. So that means 2 amino acids have been linked together before this one. So like we talked about in the movie, there's the second amino acid, there's the first amino acid. What does this amino acid have to be? What's the codon? Not the anticodon, what's the codon?
AUG? Oh.
Number 2.
Oh, number 2. That's glutamine.
So here, this is codon number 1, number 2, number 3, number 4, number 5, right? So everything we've already talked about, the transfer RNA currently in the peptidyl site is this third codon. It's carrying this amino acid. But this is the third amino acid in the protein. We've already translated the first 2 codons. So we can also work our way backwards. So if we go here, this is our polypeptide chain. This is the primary sequence of our protein. We've only made 3 amino acids so far, but we know what they are. So this is the first one, this is the second one. So this is the second codon. So what does this amino acid have to be? Is it glutamine? I don't even know. What happened to our... So the codon is 5' CAG. So CAG. Glutamine.
Why is it CH?
Oh, it's G.
What?
I thought we were doing that one.
Are we so? You thought we were doing what?
I thought we were doing that coding that CAG.
So is that a CAG not a glutamine?
So it would be
It's
G.
glutamine.
Okay, you can
Wouldn't it be G or
So
C?
you because it starts
That's
with
an L.
That's just its anti.
That's just GLN.
So this is where
I'm so confused.
I love the fact you guys are confused right now, because it's a really good point to illustrate, right? These charts only show codons. They do not show anticodons. So now is the time to be confused about that, right? So next week you won't be. Because I can give you, and I guarantee you a question like this, like a messenger RNA with 1 codon, and that's it. And you can tell me the anticodon. You can tell me what amino acid is attached to that transfer RNA. You can tell me the primary sequence of the protein all the way up to this transfer RNA, which is what we're doing now. Excuse me. You can also tell me, which we haven't talked about yet, what the next amino acid and transfer RNA is going to be. There's a ton of information that we're talking about right now. So I'm glad you guys brought that up. These charts, and the chart I'll give you, they only show you codon sequence, 5' to 3'. Okay? So if we look here, we're talking about the second codon, which is this one right here. It's a CAG. So we find that on our chart, that's a glutamine. Right? So if we go to our first codon, what is it? What is it always?
It's always MET something.
The star codon's always an AUG, but it's always methionine.
Fine.
Yep. So this is methionine.
Oh, so for these things, you follow the actual, you don't, the DNA, you don't follow the RNA.
So there's no DNA at all on this board. This is, because there's all U's, right? This is our messenger RNA, right? Yeah. No, that's why, like, you know what? Let's make this harder.
No.
Not harder, let's make it longer. So we can practice more. What's another good codon? We're gonna do a... So now if we look here, there's our start codon. There's codon number 1. There's codon number 2, codon number 3, and codon number 4. Each codon is going to be codes for an amino acid, right? So currently on our 1, 2, 3, 4th codon, we have a transfer RNA sitting there on the ribosome. It's the one that's currently being translated. We already talked about how the anticodon of this transfer RNA binds to the codon sequence of the messenger RNA, and if you're a transfer RNA that has this anticodon, you have to carry a 3'. Okay? So now let's work our way back. Let's go, just for fun, to the second codon, which is going to code for the second amino acid. So if you look at the messenger RNA, the codon sequence is 5' moving this way toward the 3'. It's 5' CGC. That's a messenger RNA codon, so we go straight to the chart, right? What amino acid does that code for?
Arginine.
Is it arginine? So there's an arginine right there. Okay?
Yeah, that makes sense, sort of.
That's why we practice, right?
Adds on.
So this is our third codon. It's a CAG.
So we have.
This is our third amino acid. All right? So if we look at our codon here, which is a CAG, go to our chart. What amino acid does that code for? Glutamine. And then we go to our first codon, which corresponds to the first amino acid. And what is AUG always?
Methionine.
Methionine.
Yep. Isn't that the start codon then?
Yep.
Okay. And UGA is the stop codon?
Always the start. There's always the start codon. Always. Okay. Let's complicate this a little bit. So let's say that we back this up in time. A transfer RNA had to come to the ribosome carrying this arginine amino acid. What is the anticodon of this transfer RNA have to be if it's going to bind to CGC?
GCG.
It's got to be a GCG.
Alanine.
Is this the 5' end of it or the 3' end of it?
That's the 5'.
5'. Has to be anti-parallel, right? The 5' right here, so it goes 5', 3'. Remember a tRNA, we draw them like this because that's actually what they look like, but it's one single strand of RNA just folded up. So what that means is so if you look here, if this is the codon, 5' to 3', and it's, let's take it out by itself. So this is just taking this one codon out of this messenger RNA. Here's 5' end of it, then it goes CGC 3'. That's all I drew right there. The only RNA molecule in the world that's going to bind to that, so this is our codon, the only anticodon that's ever going to bind to that
Is alanine.
is going to look like this. Is this going to be the 3' end of it or the 5' end of it? That makes this have to be the 5', right? If this RNA nucleotide in the codon is a C, what does this have to be in the anticodon? G. So it needs to be 5' GCG. 5' GCG. So that's right.
So you can just read it backwards every time, pretty much.
Yep.
Make life easier.
Yep. There's nothing wrong with making life easier. So now the next question is this. What is going to be the anticodon sequence of the transfer RNA that comes in next into the A site?
AGU.
So it's going to be, what's here? A. And then here. Okay, so the anticodon on this transfer RNA, it's the one coming in next into the A site. It's going to go 5' UGA because that's the only nucleotide sequence that will bind to this one. Now here's the crux for this part of the question. What amino acid is this transfer RNA carrying? And before you answer that, are you going to use the anticodon sequence or the codon sequence on that chart?
Codon.
Codon. So what amino acid is this transfer RNA carrying? Serine, is it a serine? Yeah. AGU? Yeah, it's just,
Siri.
yeah. Serine.
Serenine.
Siri.
Serenine. I like serenine better.
Just make one up every
So that's the last part of this question. Like then we watched that movie, right? Once the ribosome is done here, it ratchets down one amino acid, so that or one codon, so that another transfer RNA has to come in. But which one, right? And it has to be the one whose anti-codon binds specifically to the codon. That's how the ribosome knows which amino acids to link together. Because remember, all a protein is a sequence of those amino acids, all these 20, in a particular order. That's all a protein is. But if a protein has one of those amino acids wrong, then the protein may not function. Or it could be awful. It could be sickle cell anemia. It could be cancer. Like you cannot make mistakes doing this, right? So if I give you this picture here, again, starting out with nothing but a sequence of messenger RNA and a completely unlabeled transfer RNA, you should be able to tell me what the codon is, what amino acid is attached to that particular transfer RNA. Then you should be able to look at the codons that have already been translated and tell me all these amino acids in the right order, right? And then the last thing I want you to be able to do is look at the next codon, the one that's in the A site, and then take an unlabeled transfer RNA, tell me what its anti-codon is, and make sure you label the ends, and also what amino acid is attached to it. I guarantee you that question. What's that?
Can we have a practice one?
Yeah, sure. Yeah, absolutely.
Is there a specific amino acid for the stop codons, or is it just clip it right there?
So that's a good question. So this is our last regular codon, right? So the last part of this is the ribosome has to know when to stop. So there's 3 stop codons. Why is there one start codon and 3 stop codons? I have no idea. Like it just doesn't make any sense, right? So somewhere up here is a UAA, UAG, and UGA. So there's 2 of them. And then where's UAA? Right there. So UGA, UAG, and UAA. When you hit any of these 3, right? This is a stop codon. Do stop codons code for an amino acid? Yep, I almost wrote condom right there. Stop codons do not code for an amino acid. Period. Right? When a ribosome ratchets down so that there's a stop codon in the A site, no amino acid is going to be formed. No transfer RNA is going to bind to, has an anti-codon that binds to a stop codon. What happens is other proteins, these aren't proteins, these are RNA. Proteins bind to that whole stop codon and the whole thing falls apart. Okay? So I want you to, I realize this is kind of a bit of overkill, but we're gonna watch that video again. Oh my God. After talking about it.
Can I just ask something real quick? So
Raising
the
Cane's
first
full
strand,
menu.
that's where
That
you
looks
get
delicious.
the
That's what he
amino
wants to
acids.
say.
And the other transfer
Okay,
RNA is
that
where you
sounds
get
really
the
good.
I want a whole like
For
Exactly.
cup of like cane sauce.
So based on her question, like when you look at what's actually going on with translation, right? So there's going to be a messenger RNA right here. There's a ribosome translating it, right? Small subunit, large subunit. How many transfer RNAs are going to be right there as well? Thousands, millions, right? Because to translate a protein, you have to have a transfer RNA that has an anti-codon that binds every single one of these. There are many, many, many different transfer RNAs, right? But if you have an anti-codon that binds to these 3 sequences, then you are going to be carrying a valine. That's the link between amino acid sequence and messenger RNA. And we could absolutely do a practice one. Okay.
Wow.
But this is cool, because this is how life works. This is how living things make their proteins. That's how you take a code and you build molecules that can read the code from the DNA, which lasts the whole life of the cell, which can be your entire life, right? Make a molecule like mRNA that doesn't live very long, but lives long enough to be a set of instructions that completely different organelles, using the help of different molecules, can use to build the things that make you up and control how you work. That's amazing. It really is. That's like life, right? So.
You said all the tRNAs are inside the ribosome?
There's like a, so if you wanted to have like a kind of a size, so like here, if this is our ribosome was like this big, I mean it's not, let's just say it was, then we would have our messenger RNA and there would be millions of like microscopic little transfer RNAs. And what happened to the transfer RNA that was here, right? That transfer RNA lost its amino acid. It came in holding this glutamine, but that was transferred to the transfer RNA in the P site. So this particular tRNA that's right here, right? So if we were to draw it, it's been used up, right? But we know what amino acid it wants to be carrying. It wants to carry a glutamine. And so wouldn't it be great if there was an enzyme that would recharge it with the glutamine? And there is. There's a whole family of enzymes. It's a mouthful. They're called aminoacyl tRNA synthetases. And what they do is take empty tRNAs and stick the right amino acid right back on them. And then they can come back. They're recycled. And how do those enzymes know which amino acid to put on an empty tRNA? They read its anticodon. Okay, are we good with this for now? Okay.
I actually have one more question.
Yeah,
So,
for sure.
like, the possibilities of like the different codons has to be like endless, right? So, for example.
No, but it's not though. Those are all the ones there are.
Kind of like, it goes on for really long, no?
Yeah, well, some proteins, like 100 amino acids long. There's one in our muscle cells called titin that's like tens of thousands.
So my question is, what if there's like 2 start codons before it hits a stop codon? Or is that a mutation? Or is that never gonna happen?
No, that happens. I wish they'd let me teach like a 400 level genetics class. We could talk about that. So both of the start codons will be used, but the one downstream is only used like maybe 5% of the time. So we do that. Or we say we only have 20,000 genes, which is totally true. But what we don't talk about is that when a gene is transcribed, you can make a messenger RNA that can be translated in different ways. So sometimes that actually happens. In bacteria, it happens all the time. We'll talk about here in a little bit. But in us, it happens too. So sometimes we use one other start codon too, super rarely. So it's only for proteins we need a tiny amount of, right? If you think about this, so small tangent. So if you had like a 5' untranslated region and then a start codon and in a long coding sequence, another start codon, and then eventually a stop codon, let's say this made a protein that you needed a lot of, but this made a protein you'll need a little bit of, then this is a way more efficient way to make it. If this only gets translated, so if the ribosome skips this AUG like 5% of the time and then starts at the second one, well that's great if we need this protein in really small amounts and this one in large amounts. So our body does that all the time. Yeah, okay. So are we good with transcription and translation? Okay. I'll make a note or I'll forget. I'll put a practice problem on there. Okay. Where are we at?
So is that the end of, translation?
Yeah, so we're done with translation. I'll put a practice problem out there, but we will talk about it again. Okay. So moving on from there, there's nothing new on this slide except for one thing. When we've talked about gene expression from a eukaryotic point of view, we've had the DNA in the nucleus, right? So RNA polymerase transcribes the gene, makes messenger RNA, and then that messenger RNA has to leave the nucleus and go out into the cytoplasm where the ribosomes are to be translated. Do microbes have a nucleus? Do they have any membrane-bound organelles?
No.
Nope. So there is no reason that translation of a messenger RNA in a prokaryotic organism has to wait for transcription to be over to start. Okay? That's what this is showing you here. So one reason that microbes can grow and divide so fast is because if this is the sequence of a DNA gene, right? RNA polymerase is transcribing the messenger RNA right here, and it's not even done. This is the 5' end of the messenger RNA, but the 3' end is maybe down here somewhere in the DNA. So RNA polymerase is still transcribing the gene, right? So maybe here it's only 5% of the way done, but the 5' end of that messenger RNA is sticking out away from the DNA, and there's no reason ribosomes can't bind to that and start translating the messenger RNA before the RNA is even completed. And that's what you see here. This is an RNA polymerase that's ahead of this one, and this one is ahead of this one, and this one's the one in front, right? This RNA polymerase may not even be done yet. It may be nearing the end of the gene, so the messenger RNA is really long, especially compared to this one, but look how many ribosomes are already translating that RNA. So because prokaryotic organisms do not have a membrane-bound nucleus, their DNA is not separated from their ribosomes, the messenger RNA can be translated while it's still being made. And so bacteria can make proteins incredibly fast. And this is just electron microscopy. That's the DNA, right? That little line right there you can see, that's that. These are the RNA polymerase. And then there's the long chains of messenger RNA covered with ribosomes that are all translating toward the 3' end. Okay? That's one reason microbes can grow and divide so fast. Okay, so this is going to be a brand new concept. Maybe. Has anyone here ever learned about operons before? You covered that in your prereq? Anyone? Okay. So the reason I'm drawing this up here is I want to remind you that everything we're talking about, at least initially, is DNA, right? So operons are a sequence of DNA that have a bunch of very specific parts, right? So the way we usually draw different elements of a DNA molecule is just by drawing boxes, right? So when we're drawing the boxes and giving the boxes names, just remember that it's still just one long DNA sequence, right? So this marker's dying already. Remember when we talked about metabolic pathways, right? So if you start out with a substrate, let's say we're starting out with a substrate and we're ending up with a product, but we need 3 different enzymes to make it happen. So enzyme 1 is going to give off a product, which is the substrate for enzyme 2, which is going to give off a product, which is going to be used as a substrate for enzyme 3. Is this pathway going to work if I'm missing enzyme 1?
No.
Is it going to work if I'm missing enzyme 3?
No.
Right? So here's the thing. A lot of times in a biochemical pathway, if you need that pathway to happen, you're going to need all of the enzymes. So you either need all of them or you need none of them. right? And so because of that, bacteria do something that we don't, right? They have a system called an operon where they can make all of the proteins that they need for a metabolic pathway all at the same time, okay? So they either make all of them or they make none of them. So we're going to talk about a specific example. So the whole point of an operon is to control when you make specific proteins. So these structural genes right here that we're just calling Z, Y, and A, you can think of those as the enzymes. So let's just rename this. We're going to say that this one is Z, this one is Y, and this one is A. So if we need Z, we need Y, we need A, we need all of them. So we're going to make them all at the same time, or we're not going to make them. These are proteins, but how do we control when they're expressed in the DNA? And the answer to that is an operon. So an operon consists of all of the structural genes that code for the enzymes you need in a particular pathway, plus an operator, plus a promoter. That's the operon. These are new words: operon, operator, and promoter. We'll talk about specific ones here in a second. And then somewhere away from the operator is what's called a regulatory gene. Okay? So let's talk about a specific example of when you would have an operon. There's many, many different kinds of operons, but one of them is called the lac operon. Lac is basically abbreviation for lactose, right? So that is, what you're looking at here is a super generic picture of an operon, but let's talk about how it's used specifically. So this is the lac operon, right? So let's talk about how it functions and what it's for. So these are the structural genes. So this is Z, this is Y, this is A, right? And remember, we're just naming sections of the DNA molecule. This is every little box we're going to talk about, nothing but a named sequence in the DNA, okay? So, quick aside, you don't need to know this, but these structural genes for the lac operon all have to do with enabling a bacteria to use lactose as an energy source, right? So if there's no lactose around, is it worthwhile to make these proteins?
No.
No. So if a little bacteria is cruising around and there's no lactose, sugar, right? If it's making all of these proteins from the structural genes, but there's no lactose, that is a royal waste of time and energy. It's a waste of ATP, it's a waste of nucleotides, it's a waste of amino acids. And so that bacteria could fall behind the ones around it, get outcompeted, and it's going to die. So bacteria do, and us, we do not want to make proteins if we don't need them. So if there's no lactose present, we do not want to express these genes. Okay? What you don't need to know, this is beta-galactosidase, right? It's a permease and a transacetylase. What it basically does is make this big pore in the cell wall that sucks the lactose in if it's around, and then the beta-galactosidase chops the lactose up into monosaccharides bacteria can easily use. Basically, these structural genes help the bacteria get and use the lactose if it's available. But here we're going to say no lactose is present. So the whole point of all of this stuff on this side, this is going to be different over here. Everything over here is going to happen if there's no lactose present. And the point is, we do not want to express these genes. It's a waste of time. There's no lactose present. We don't need the proteins to break it down, period, right? So this section here is called an operator. An operator is where a repressor protein binds. So if repressor proteins bind to operators, as we're going to see here in a second, that prevents transcription of the structural genes, okay? Now, upstream of the operator is what's called the promoter, which we've talked about a little bit. A promoter is where RNA polymerase binds. And then, just to signify that this is the end of the operon, I'm going to draw a little bit of DNA here. This could be 1,000 base pairs, could be 10,000, doesn't matter. But somewhere else in the chromosome is a repressor gene. So, and in the case of the lac operon, it's called I. It's called lac I. So lac I stands for lactose inhibitor, right? You don't need these genes being expressed because you don't need those proteins, so we're going to shut the whole thing down at one time. So if there's no lactose present, then we're going to take the repressor gene, the lac I gene, and we're going to transcribe it. This is lac I mRNA, and then we're going to translate it, and we're going to get our lac I repressor protein. What do you suppose the lac I repressor protein does once it's made? Binds to the operator. Operators are where repressor proteins bind. So when the lac I gene is expressed, we make our lac I repressor protein. Our lac I repressor protein binds to the operator. So now what happens is RNA polymerase just wants to do its job, right? So RNA polymerase comes and binds to the promoter, because that's what RNA polymerase does, and RNA polymerase gets everything it needs to transcribe all of these structural genes, but the lac I repressor protein is sitting right there bound to the operator. It's like getting in your car to drive out of here, and someone parks right in front of you. You're not going anywhere, right? So if the lac repressor protein is bound to the operator, RNA polymerase can bind to the promoter just fine, but it can't transcribe any of the genes. So we don't make messenger RNA for these genes. They don't get translated. We don't get these proteins, and that's exactly what we want to have happen because there's no lactose present. So why make them if we don't need them? Okay? Now I made this thing looking all funky. Like, why did I make it look like that? Right? And the answer to that is the lac I repressor can do one other thing, right? So we're going to switch gears and talk about that. So this is my terrible drawing of the top half of this picture, right? So now we're going to switch gears and say lactose is present. Okay? So lactose is our little disaccharide I think we've talked about before, right? Lactose is a glucose and a galactose bound together. So if lactose is present, some of it's going to make its way into the cytoplasm of the cell, and what's going to happen is that lactose is going to bind to the lac repressor. So the only way lactose can bind to the lactose repressor protein is if lactose is present, right? So if lactose shows up, now the bacteria is like, sweet, I have sugar I can use. Let's put it to work. Let's use it to make ATP, but to use that lactose, we've got to have these enzymes. That's why lactose turns this operon on. And the first step of turning it on is to take the lactose, which looks like this, it's a disaccharide, it binds to the repressor protein. When lactose binds to the repressor protein loses the ability to bind to the operator. Now, is there anything stopping RNA polymerase? Nope. Transcription starts. right? Now we're making messenger RNA for Z, Y, and A. Now we're translating that into protein Z, Y, and A. We put the sugar to use, right? Now eventually we're going to run out of lactose. When we run out of lactose, there's not any left to bind to the lac repressor protein. So what does it do? It binds right back to the operator and shuts this operon down. And because nothing lasts very long in a cell, right? Proteins don't last that long, messenger RNA doesn't last that long. The only thing that lasts for a long time is the DNA. So when the lactose goes away, now the lactose isn't available anymore, so that when the lactose leaves, the repressor protein binds right back to the operator and turns that whole operon off. Okay? So that's how a microbe uses an operon to make sure that it can turn off or turn on the expression of multiple genes at one time. So it only expresses the genes when they're needed and shuts them all off when they're not needed. If a bacteria didn't have an operon, then Z, Y, and A would be far apart from each other. So you'd have to have 3 promoters, and you would have to have 3 operators, right? And you would have to have 3 different repressor genes. But why do that? Because you either need all of the proteins or you don't need any of them. Microbes have pared their genomes down to as small as they can possibly get away with, because the smaller your genome, the simpler you are, the faster you can divide. Okay, any questions on operons? Okay, so I should be able to tell you, okay, Lily coli is cruising around, lactose is present, draw the lac operon in the conformation that it's going to be in. Or if lactose isn't present, what's it going to look like? Kind of thing. Okay. How have we been here an hour already? Okay. So I do want to make time to go over the exam. Keep putting that off. So let's call it here for today. So we'll finish this chapter up on Wednesday. So it looks like exam number 3 is going to be chapter 8 and chapter 9. And I'll give you guys a practice problem of the one that I'm gonna, like a guaranteed one I'll give you for everything we talked about this morning. If you can answer that question, like what we went over earlier, like you understand transcription and translation, right? There's a lot there to put together.
So if you say it's not present,